everyone, and welcome to the latest interview in the Down the Rabbit Hole series. Today we are going to be talking about a really cool area, which is DNA data storage with James Banal. So James, if you could just introduce yourself and tell everyone a little bit about what you do as well.
I'm James Banal. I'm a postdoc currently at MIT in the Professor Hypothesis Lab. I am a chemist by training and tried to dabble a little bit with biology, and so I'm here now in MIT working on some of the wacky stuff we can do with biology, like quantum computing and the topic today, DNA data storage.
So what is your kind of main research focus at the moment, or more importantly, what is your favorite part of what you do?
Yeah, so actually what's funny is that DNA data storage, this project was my back burner project. It wasn't my main project. So the actual main project I'm working or why I was hired here was on how we can use DNA to imitate how photosynthetic systems look like, and essentially try to figure out are there like quantum effects in there. And DNA data storage was sort of this back burner project that I took over from another postdoc, and we were trying to figure out how do we solve this problem of how do we build essentially a Google version for DNA data storage. So that's how we came up. So the most exciting thing, I think, for me, being a chemist in training, is like how powerful biology is. I'm always very overwhelmed by the fact that you can do so much with biology. As a chemist, we have this mixed chemicals, but with biology, it's like biology, like you have another hand that can do a lot of stuff for you, essentially, because you have bacteria and stuff like that. So I think that's the most exciting thing for me, being able to re-engineer biology for some unusual purpose. So I think that's the most exciting stuff for me.
What has it been like for you? Because you said obviously you're a chemist by training. So what has it been like trying to kind of go into biology? Because they are quite different. Obviously, I know they combine like in areas, but what has that kind of been like for you? Because especially you've got to kind of learn on the job as well.
Yeah, definitely learn on the job. It was a struggle at the beginning, simply because like the language I use is different, and the language of chemistry and versus biology is sort of different, and the methods are different. I've also worked a little bit on physics, so trying to figure out one of the craziest things is you're talking to computer scientists, you're talking to biologists, you're talking to a whole different physicists all of a sudden, and you have to somehow bridge the gap between the vernacular between the different areas. And so it was quite tough in the beginning, but you like I said, you got to learn when you on the job. And so that having the chemistry background did help, but it was a struggle in the beginning. Yeah.
I mean, as I mentioned at the start, we're going to be talking about kind of DNA data storage, but if we just kind of take a step back, I think when people think about nucleic acids, we often associate it with kind of the building blocks of life, and it makes us and all that kind of stuff. But I think over the past year, with the kind of mRNA vaccines and everything like that, I think we have suddenly realized how much more we can do. Like we can use these kind of nucleic acids for kind of other applications, and obviously one of the most exciting ways is like through like therapeutics, obviously with the mRNA vaccines. What are some of the other kind of ways in which nucleic acids are being used within this kind of therapeutic area?
Therapeutic area. Actually, our lab is very active in that, and so you one thing that you can think about is the way we think about nucleic acids, basically like beads on a string, and you basically have 4 different beads of different colors. But in reality, you can also think about it as a polymer, like a chicken wire that you can sort of bend in different ways. And that sort of thinking actually led us, a pretty smart fella named Adrian Seaman in NYU discovered a way to how we can structure DNA. Using some of the motifs like found in biology, but he sort of created this novel idea on how we can structure DNA in different shapes, And then Paul Rotherman and Caltech developed a very cool way to fold or fold DNA. So that's what we call DNA origami. And they can like structure different shapes and sizes of dimensions of DNA nanoparticles. And when you think about that, it's like it is a scaffold, and so that's where my work on photosynthetic systems was like, can I mimic some of the structure and nanoscale organization of stuff using DNA as scaffold? So for a therapeutic sense, you can think about it as like they look like viruses. So we actually made this a DNA origami that looks like a virus. Like it looks like a icosahedron. If you play a D&D game, you can actually make some of those dices that look like into a DNA origami too. And then because it's DNA, you can synthetically modify at like the base resolution, because all the advances we've made in DNA synthesis. You can present antigens, control the different orientations of those antigens, which actually can create a whole different spectrum of, say, immune response, or you can probably deliver CRISPR, so I see it as what people don't know yet is that area that's burgeoning in the field is how we can use DNA origami as a therapeutic vehicle, essentially, which is, we've heard about liposomes from mRNA, but what if we can structure the mRNA and just essentially deliver it to the cell? And I think that's the most exciting thing, thinking about that as a vehicle. So that's the alternative way. And there's a lot of different ways you can also use DNA that it's not yet therapeutic, but also, I mean, we're going to talk about DNA storage later on, but I think the other exciting thing is how can you use it to spatially position stuff on the nanoscale? Something that I'm very actively working on in the lab is because the ability to structure DNA very, very, precisely because of this Watson-Crick-Franklin base pairing that allows us to program exactly the desired interactions, and then the way we know the mechanics of DNA. And so we can have this like 2D structures that you can use to build up materials. And there's a lot of activities in that, trying to build electronics from the ground up. So that's actually one of the other exciting besides therapeutics.
You don't realize like how much kind of physics and kind of like engineering comes into like DNA, because I feel like I kind of know more of like the more clinical side, but you actually don't realize how kind of complex it actually all is. And kind of from your kind of perspective, what are some of the challenges within this kind of like therapeutic area?
Yeah, there's a fair bit of challenges, right? So I mean, one of the challenges is, I mean, we could start with like, is it scalable, right? Can you manufacture at a large scale? And there's been work in our lab that we can show, and that we can create larger. We can you basically create nucleic acids that essentially are the scaffold. So how DNA origami works is just take a really, really long strand, like several thousand of bases, and you essentially fold it using small strands of DNA. So the big strand, the long strand DNA, that's not very easy to make, and you have to have specific sequences, right? And so the innovation in the lab is how can you make that in milligram scale, almost nearly arbitrary sequence specificity on like the whole 10,000, from anything from 1,000 to 10,000 base pairs using bacteria. And so with that sort of problem, the scalability issue sort of solved, then the next challenge would be how do you actually scalably attach stuff on the linear origami. There's a lot of chemistry that's been involved in that. There's a lot of the click chemistry that people are probably very familiar about. And so a colleague in my lab has actually developed a way to functionalize DNA nanoparticles using click chemistry, and demonstrated that in a very beautiful way how you can put different stuff on the DNA origami. And then the next challenge then is the stability. Now, stability is a little bit challenging to I think it's very early stages, but because it's DNA, and we put it in the body, there will be it might be degraded by some nucleases, or I mean it could be hidden somehow, or yeah, so there's a lot of different things we don't know yet about DNA or about these DNA nanoparticles for delivery for therapeutic purposes. Yeah. But it's essentially, yeah, those are like, I think the biggest challenge. I think the next, big challenge, the big delta we need to make huge effort on is stability, and the next step is like what therapeutic targets do we actually go after once we know how to make it stable.
No, it sounds really, really exciting, but kind of one of the areas that I'm most obviously excited in is kind of DNA data storage, because I actually didn't even realize this was even a thing until like last year, and my mind was blown. Like I was like, no, this can't be real. And then I obviously did loads of research and I was like, no, it is. But for those who don't know, kind of, would you be able to summarize what this actually is?
Yeah, I mean, in a nutshell, what DNA data storage is essentially converting the zeros and ones, the binary digits or bits of digital data, into the 4 letters of DNA, the A's, C's, T's, and G's. Now, that's an oversimplification with all the process involved, but essentially at the core is you're using the beads of molecules of strands, the beads in a strand to store your digital data. And that has a tremendous impact on how you can actually store a lot of data in a very tiny space. So that's why there was a lot of motivation to think about digital data. But what really probably good is to go like maybe take a step back and like why people are actually working on this is actually because the amount of data we're generating just is just immense, and we're gonna run out of space to store them. I mean, with the internet and TikTok, especially last year, yeah, it's insane how much data we're gonna generate. I mean, it's just gonna grow exponentially.
Yeah, but what are some of the kind of like processes involved? You can go into like a bit more detail about how is it actually done? Like it's just obviously it's really, really difficult to explain verbally without like kind of seeing it. But yeah, like what is the kind of like process to actually being able to do this?
Yeah, so for example, you take a picture of a cat and you want to convert the DNA. So the process essentially is you convert that to essentially to the DNA sequence. Now there's a lot of technical details though, but essentially is that you convert those into, because we are limited in amount of the length of strands we can synthesize because of current limitations of DNA synthesis. And so you need to figure out a way how to segment those in multiple strands, add indexes so you know actually the order of how they should be read, and then you add addressing barcodes so that you actually know what file it is. Is it a picture of a cat? It's a video of a TikTok dance or something like that, and so something like that. So that's how we get to the DNA sequence. Now the synthesis. So the synthesis, there's a lot of different ways. One of the most common ways is to use a microarray, which is Twist Bioscience is really good at. So essentially, what it is you grow the DNA polymer from the surface 1 letter at a time. And they can do that in a very parallelized way using a lot of different technologies. Actually, a lot of it's from printer technology. Most people don't think about it. It's like the inkjet printing. And so like growing those strands 1 letter at a time is what makes it that's how you synthesize from sequence to the actual physical DNA. And you store them. And once you store them, the way to access them is the current way that most people did, is use polymerase chain reaction, because essentially you basically put primers, which I think about it as like just a bunch of Velcro molecules that would stick on only specific sequences. And they would, when you it primes, it amplifies the DNA, you take many copies, and everything that doesn't have those primers won't get amplified. And that's what you put to a sequencer. And then there's 2 flavors, both of them from the UK, and one of them is Illumina. And the other one is nanopore sequencing. And you basically read the sequence and then you convert it back to the file, digital file. Now there's a lot because there's so many steps. There's a lot of things that can go awry. So like one of them is when you actually try to synthesize it. So there's still errors when you write it. And so there's a lot of different ways to mitigate that. There's a lot of error correcting codes so that, you know, even you can win because you don't synthesize only one molecule of DNA. You actually synthesize many molecules of the same strand. So you can win by that, but then they will have some errors. So you need to figure out a way how to correct for those. So you have some error correcting codes that you have to put in. So essentially, you need to add that, and all those error correcting codes also help downstream. Say, for example, you lose that one strand that you're important, and so you can sort of figure out the pieces that you to fill in those blanks once you actually start to decoding it. So there's a lot of technical details there, but essentially that's currently how it's like perceived to be done. Yeah.
And what are some of the benefits of using DNA? Like, why have we chosen to use DNA to help us store stuff?
Yeah, so I mean, DNA is pretty cool material. I actually was super impressed by it. Well, one is like we're storing bits of data uh, on the molecular level. We're talking about roughly 30 atoms. Each nucleotide is probably like 30 to 20 to 30 atoms. And if you think about current devices like in your phone, for example, that's like a lot of atoms. It's 1,000 atoms easily for just for a few bits. And so that's really one of the things. That's how the density is, right? So that's how you get the density down from silicon that's big to something that's really tiny. The other thing is the longevity of DNA. So DNA is just a sturdy molecule. I don't know. We've been able to extract nucleic acids from fossils, and those fossils, by the way, have experienced the harshest environments you can think of, and they still survive. The data still survive. Think about your phone, or I don't know, your disk drive. You put them in that same environment, they won't survive that. They're basically gone. So like this durability and longevity of data that can be retained is essentially it's very unique to DNA. The other thing the other thing that I actually really like is the idea of you can create a lot of copies very quickly. Like I've talked about PCR, but there's other, like you can use bacteria. It can make, and we're not talking about just 2, 3, 4, 5 copies, right? We're talking about trillions of copies in the order of Avogadro's number, 6 × 10⁻23, right? And so that's I think that's the most one of the most compelling things. And the reason why you want to do that is because if you want to preserve data, you want to have multiple copies of that. Maybe put somewhere in Mars. Ask Elon to shoot some of our internet in Mars. So all the tweets from Trump or whoever gets preserved forever, and put it everywhere. You can actually do that with DNA because it's durable. You can make a lot of copies. But you start to do that with other devices, then you can't really just do that very easily because they won't be able to survive. And making copies is just too much. The other one's computation. And that's actually something that our lab is very interested in, is because the way we think about computation is like it's very two-dimensional. You move electrons around and something like that. With molecules, you're taking the fact that these molecules are jiggling in solution, trying to find each other through diffusion, and that's really fascinating because you basically have the ability, in theory, if you do it right, you can have number of CPUs in the order of Avogadro's number in a very tiny space. And it's hard to find something like that in silicon. And so that's actually, I think, to me, it will be the biggest reason why DNA is going to be probably the next data storage to look at.
I know obviously that the field is only kind of starting, but what is like our current, what are our current capabilities in terms of what we can store, like what are we able to store at the moment, and also what we hoping to be able to store in the future? Like how much are we going to actually be able to store on this, or is it just like we're hoping endless?
Yeah, so I mean, currently, because of this exorbitant cost of DNA synthesis at the moment, we're limited in pictures, videos, a few clips. Someone has already done sonnets, a music file, MP3 file. I mean, we basically, someone actually has put a computer virus too, and a whole operating system. which is actually quite exciting because now like we can literally preserve anything. And so we can literally preserve anything now, in the sense that all those files can be, in theory, converted to DNA. In practice, the only limitation to the cost of DNA synthesis. What kind of and the vision that we wanted to, what we published on recently was, can you actually make it searchable? And that's something that I actually really think is going to be important, because here's why. The internet revolution wasn't, didn't fly off because of our ability to, Google didn't, their value didn't rise because of the ability to write data on the internet, right? It wasn't that. Their value was being able to search stuff. And so I think the random access bit is something that we worked on and still working on, how to make it faster, better. It's essentially like Google still working on their search engine. And so we wanted to do the same thing and reach that level of complexity and how to search stuff. And that's with the random access, because you don't, in theory, if we store the whole internet on DNA, you don't want to sequence the entire DNA just to find a picture of a cat. That's just not going to be a practical thing to do in terms of time and money. And then finally, on the sequencing side, there's a lot of challenges there too. It's still not cheap. It's not yet. We're not talking about $10 per run. No, it's probably still $100, something like that, per run. And so, not counting the steps to actually make it compatible with the sequencer. And yeah, that's basically in terms of cost. Speed is another problem. Writing data on DNA is still slow. I think we're still in the I think we're in the megabytes now. Who knows? We don't know from other companies, but I think we're close to having to megabytes per day. I'm not talking as per second or per day. It's just horrendous to think about. So we're not there yet in terms of that writing. See, the search thing is also slow. We're at gigabytes per second now. That can be done, but if you're going to look at an exabyte, the whole exabyte, that's going to take you forever. So we need to make that faster. Finding the needle out of a haystack needs to be faster, terabyte. And I think we can get there actually a lot easier compared to the other problems. And the nanopore, this nanopore and Illumina sequencing, the reading is also still quite slow. So we need to make that faster. There are a lot of obvious ways to make that faster that I think these companies are working on.
Yeah. You and your team recently developed a new retrieval technique. Would you have to just explain a little bit more about kind of what that is and why you did it and everything like that?
Yeah, the inspiration is actually Google. So how Google manages information is basically have a file, and underneath that file is a lot of metadata. Metadata that, cat is orange, cat is domestic, cat is from, from New York or from Cambridge, UK. And essentially those metadata is how, for example, you want to say orange cat, that's how it actually populates that data set, because it matches the language or the query that you put it in the query box. And so that's exactly what we wanted to do is, can we implement the same thing? And one of the biggest things that we had to figure out is, how do we separate the data file, the main data DNA sequence, from the metadata DNA sequence. And currently, right, I mentioned about the PCR. So the metadata and main data are all in the same strand. The problem with that is, as you scale to the exabytes to, yottabytes, what's going to happen is that you'll have some challenges on how many barcodes can you actually use. And you have a limited length of DNA, so your real estate is very limited. So what you have to figure out is, for them, is how they're going to figure out is how many barcodes can we use to uniquely identify them. So we wanted to separate that problem altogether, because we want to attach as many barcodes as we want without the restrictions of DNA synthesis. And so that's where the encapsulation came in. And I'm a very bio-inspired researcher, so like I mean I'll be doing photosynthetic stuff. So why not take how the cell actually manages our nucleic acid? Like the cell puts the main data, basically the DNA data, our genome and nucleus, and outside is like where the all fancy signaling happens. And so can we do the same thing synthetically? And essentially that's what we did, is just encapsulated in silica, which is a very hardy molecule. It's glass essentially, and essentially put multiple barcodes in the surface, different barcodes combinations. In theory, and we took from this pool of 240,000 DNA barcodes that we are orthogonal, so we don't have to do the computation to do that. And then if like you think about that, 240,000, say just 10 combinations, or maybe 20 like combinatorial combinations of those barcodes, can actually get to like combinatorial explosion. You can basically, in theory, store every atom in the observable universe with that number of combinatorial combinations, essentially. And so that was really powerful idea, because actually that's what's the problem for the whole data storage field in terms of random access, scalable indexing. But we want to take it to the next level and actually build a file system, because the analogy here is like, it doesn't matter if you can read and write data, if you can do anything with it other than just read and write, if you can compute it, then that becomes the most powerful. And what we demonstrate essentially is like, we can do Boolean search logic. Basically, how would you search something on Google, but now on DNA? And we've done it experimentally, not theoretically. So initially we had a lot of issues with it. I had to figure out a lot of different stuff to make it happen. But then when we figured it out, that's probably like when I first did the first search, I was like so happy that actually we made it a reality, because everyone thought it couldn't be done, but here we are.
That's really, really cool.
Yeah.
But I mean, say if this is all like, it turns out that sequencing becomes really, really cheap. We're able to synthesize DNA really, really quickly and everything like that. How do we then make that accessible to the everyday person, if that makes sense? Like, how are people going to use it?
Yeah, I mean, there's a lot of models for this. I mean, the Dropbox model, you essentially just put your archival data. So one thing I should say is that will never be DNA readout will never be faster than your computer in terms of searching and that. But you can, if you allow for latency, so that it will be limited to data that's archival, basically your backups, your things that you don't actually use frequently. And there's a lot of examples of that, right? Your photos from 2012, you don't even look at that often. But essentially what would happen is that, a normal person would just upload something in the web, and then that gets converted to DNA and encapsulated and barcoded based on, say, the features. It's basically like how would Google do it, but in a more in a very different way, in a sense that you have to print the DNA and store it archivally. And I think that's the way the average person would use it. And then when they want to query, you basically have to allow, say, 24 hours to return your file. So the model for that is actually already there. It's like AWS Glacier. Basically, if you want to store something archivally using the current paradigm of data centers, then you put it there, but it's not always hot. So it really depends on like how often you will be thinking you're gonna access it. So I think that's how folks would figure out which one to do, because it's a lot cheaper to save it in DNA, because you don't have to pay monthly fees that like you would do for other services. So.
And what role do you think that kind of machine learning and things like that will play in this, if at all, kind of thing?
Yeah, a lot, actually. I mean, there's two ways to think about it: how machine learning will affect DNA, and how DNA can actually affect machine learning. So let's start with the first one, machine learning on DNA. I mean, there's a lot of interesting ways to how can we design DNA sequences that are orthogonal. Designing orthogonality is actually not a very trivial problem. Most people don't know that. That's actually there's a lot of thermodynamics involved, a lot of physics involved. Let's just put it that way. But and doing that from ab initio, from first principles, is actually not computationally expensive. So I think there's a lot of ways we can, since we know from reality there are sequences that are orthogonal, and we have basically experimental models or models that they can sort of predict that to a certain extent, and merging those two together, generating those, putting those together, and I think you can create a library of orthogonal barcodes very easily using machine learning, because it's just computationally faster than just doing it ab initio using models. And I think there are people who are working on that area. I think the other one is more exciting in my view, because how could this parallelization of reactions can actually help machine learning? And so the way if you can do searching, basically so the one thing to think about DNA is it's two molecules that bind to each other, and it's some certain strength, and the strength of that can be tuned. And so what's really interesting is that you can that's basically like machine learning, that relationships between, say, two different images or files can be connected together by some relationship. We can also do that on DNA. And so if you can do that, then all of a sudden, you can now use molecules. And the reason why you want to do that is because, like I've said, it's this jiggling of molecules that you won't be able to do in a silicon-based computer. And I think that's where it's very interesting to think about that, because one, it's more energy efficient because they're just using water and just jiggling of molecules. And two, it's I think the given the parallelizability, the amount of things you can put in that pot altogether. You just have, if you want to make bigger and bigger files, you just make a bigger pot. Not necessarily like bigger data centers or bigger CPUs or faster CPUs. So I think that's the most I think that's the exciting area. I think that's where I see DNA storage going to be very exciting in the next few years, beyond, of course, the technical issues. But I think those can be solved, and there are companies that are trying to solve that.
Yeah. It's all like so exciting, but it's also complex. I'm like, I should go back to uni and do some more learning.
Yeah.
You're involved in the startup Cache DNA. Why was this company set up, and kind of what are the ambitions and the future look like for this company as well?
Yeah, so Cache DNA was set up. The conversation about building Cache DNA was set up back in 2019. And that's when we actually had the first validation of the approach and the technology and how we gonna show it's gonna be scalable. And so we convened, some of the pioneers in the field. Well, one of them is being George Church, and Paul Blaney, who's a microfluidicist and really awesome all-around person to talk to. Jeremiah Johnson is a chemist at MIT, and my PI, Professor Mark Bata. We all convene and talk to ourselves. There's a compelling technology here, and it is a problem that I think we can solve. That's like basically the proof of concept is there. It's all about just scaling it up, make it faster, make it more efficient. It's outside of academia. We should think about building a startup. And so that's how it started, as a conversation, and we Cache became an LLC in February 2020, and that's when that's the genesis of that. But the overall vision of Cache is, if I can speak to it, is it's about indexing the world, right? So I made the analogy about Google's value was being able to index information. And I think that's where I see DNA, our technology is going to be very important, is because we actually bridge the gap between DNA writing and reading for DNA data storage. And being able to index all that information is going to be the most important thing. So imagine a scenario where you don't have that, and you have to sequence the whole terabyte, the whole yottabyte of information every time. And so for me, it's not even just that, just the ability of random access, but being able to do some computation. That's very unique to computation using the metadata. And I think that's a lot of power in there. You know, the vision is I want to be the Microsoft to the IBM. That's where I see cache being like a key player in the area. And you know, essentially making data storage a reality, helping those companies who are working on the other problems that are essentially making it useful to the average person. There's beyond, you know, just waiting for those technologies to come in to write and read. You can think about, you know, there's already pre-written DNA. Our DNA is already pre-written. There's a lot of value in taking that into you know, consolidating them, being able to search them essentially. So I mean, there's a lot of different areas now that opens up a whole new area. If you think about, you don't need to rely synthetic DNA. Why don't you just capture DNA from everyone and, you know, use it for medical purposes like epigenetics, or like trying to figure out biotech surveillance is another one. Is like, you know, what if we can track we can stop the next pandemic before it even starts, because we're able to have a library of DNA sequences from the environment, and like, oh shit, we found a bat there that has this that could have a virus that could be fatal to humans. We should probably figure that out, and, you know, maybe send Moderna, hey, here's the sequence before it even starts. Maybe we could stop the next pandemic that way. Like, having the ability to surveil the environment for those is probably gonna be important. The more we, you know, try to destroy the environments, because I think it's gonna keep happening. So I think having something like that is important.
Aside from cache, what do you think in the next few years, how do you think this field will evolve?
The whole data storage field? Yeah, I mean, I think the most exciting bit is actually having the actual. I mean, the most exciting, I think the next exciting thing is actually actual people using it. I think when we get there, I think, you know, all the pessimists and all of it is like, it's not gonna work, it's never gonna work. You have a lot of problems. I think that's gonna be the most exciting thing, is just showing them like, yeah, we made it work. I mean, it's sort of, for me, it's sort of sperm parallelism with silicon and germanium during the early days of their transistors. Like, everyone says, oh, silicon's too expensive, blah blah blah. But who won now? So I think, you know, maybe, hopefully, I'm very optimistic about my peers to able to solve and get us to a point where we could start storing information, not just toy systems, but now actually the real data from people who are needing it. And so I see it as, you know, maybe in the next 5 to 10 years, we will have the actual like a Dropbox for your data, and then you have basically just upload your file, and it's going to be in DNA. You can have a copy of it. We can set a copy in Mars if you want to, for forever, so that aliens can see, you know, your information somehow. But yeah, I mean, I think that's the most exciting area, is like that, and being able to search it. I think is the most. I think the ability to store information forever, you know, so that you never have to delete again, is the most. I think a really grand vision. Which is, I think it's going to be hard to get there, but I think we're going to get there optimistically.
Yeah. Thank you so much for joining me today, James. This field is so exciting, and I really hope that you prove all those people who didn't believe you. So thank you so much.
Yeah, thank you so much, Shannon.
Hello everyone, if you enjoyed this video, then make sure you check out some of our other videos in our series by subscribing below or going to our website, frontlinegenomics.com. I hope you enjoy.